Okay, good morning, everybody. We're going to get started. Thank you for joining us both in person and by Zoom. It's my pleasure to introduce today's speaker, Dr. Justin Drake. Justin received his PhD at the University of Iowa and then moved out west to UCLA for his postdoc training and then was recruited back to the Midwest to join the Department of Pharmacology here as an assistant professor and was recently promoted to associate professor. Justin is, I think, very well known for his work in the area of prostate cancer. He's worked extensively on the oncogene RET, understanding its function in prostate cancer and specifically neuroendocrine prostate cancer, as well as developing new ways to target RET signaling. Justin is also very well known for his taking on some technically challenging types of activities such as proteomics and proteogenomics, which it sounds like we'll hear about today, as well as identification of circulating tumor cells. He's been highly productive, publishes extensively in these areas, is highly collaborative. Most of his papers include other investigators within the Cancer Center here, as well as external institutions. So very collaborative and has been very successful with funding, including ongoing grants from DoD and NIH. So thank you, Justin, for coming and sharing your work with us today.
All right, thanks, Kaylee, for the kind introduction, and hey, everybody. So as Kaylee mentioned, I'll talk to you today mainly focused in prostate cancer, and we might dabble into lung cancer a little bit. I won't show you a lot of data in lung cancer, but I'll show you kind of the connection we have with RET in lung cancer as well. So we'll first start off, everyone has probably seen these slides a million times. Prostate cancer is the most commonly diagnosed cancer in men in the United States and the second most common cause of death. The lifetime risk of developing prostate cancer is about 1 in 8 to 1 in 9 for men in the United States. It's not all doom and gloom because about 1 in 40 men will die of the disease. So that tells you that if you're diagnosed with the disease, it does not mean it's a death sentence for you. A lot of factors play into that, including age of diagnosis, aggressiveness of disease, as well as the response to the therapies that are out there today. And the good news is a lot of the new therapies that are out there today are actually quite effective. So as you can imagine, prostate cancer is a hormonally driven disease. It's driven by the androgen receptor, androgen axis. So treating advanced prostate cancer requires androgen ablation or castration of that tumor. And that works very well in a large percentage of men, but about 20% of men or so develop resistance to this initial therapy and go on to develop what's known as castration-resistant prostate cancer. And this is the tumor that, once this tumor phenotype forms, is actually the lethal form of the disease, where there isn't really any curative option at this point, but there are systemic therapies that can go on and treat that disease. So here's an example of some of those therapies and when you might find them in the clinic. This is by no means exhaustive, but it's a general list of what we see out there. So, you can think of prostate cancer as a hormone naive or a castration resistant state. And basically in a hormone naive state, on the y-axis here, you have tumor volume and activity, which is usually measurement of PSA, which is prostate specific antigen. That's typically the marker that's typically used. A patient may undergo surgery or radiation therapy to either remove the prostate altogether or to shrink the prostate. And if it's aggressive enough, they may undergo some form of anti-androgen therapy or androgen deprivation therapy to treat that disease. And again, that works well in a large percentage of men, but some of those men will go on to develop resistance to that. And then as resistance forms, these patients will undergo more second-generation hormonal therapies to treat this disease. And then the patient kind of undergoes this roller coaster ride of response, resistance, response, resistance, and where ultimately the patient might eventually will succumb to that tumor. What we're finding now is that there's some new subtypes that are forming in this disease. So typically, prostate cancer is an AR positive or androgen receptor positive tumor. But what we're finding now is in the post-abiraterone enzalutamide, which are these second-generation hormonal therapies, we're getting an increased frequency of these other tumor phenotypes. And these are known as aggressive variant prostate cancers. And these typically lack AR signaling or even AR protein altogether. And that's occurring now in about 30 to 35% of men with advanced stage prostate cancers. So not only do we have the AR positive, now we have these other subgroups as well that are AR negative or AR indifferent. So what is aggressive variant prostate cancer? It's typically a tumor phenotype, as I mentioned, that is AR negative, AR indifferent. The transition is highly dynamic, so we think it can reverse back to an AR positive state if you find the right therapies to do that, some of which would alter, epigenetic states like EZH2. The treatment is typically platinum-based chemotherapy, and there's really no other treatment options that are really that effective. And in fact, even platinum-based chemotherapy doesn't work very well in these men. Usually the survival rate is about a year, give or take, once diagnosis happens. So AVPC can be broken down into two different subtypes. And we think there's probably even a third subtype, which I won't discuss too much, which is known as amphicrine prostate cancer, which would be kind of an AR positive state plus a neuroendocrine state. But we have double negative prostate cancer. So this is a tumor that lacks AR and lacks any neuroendocrine features. And we also have neuroendocrine prostate cancer, which lacks AR but expresses neuroendocrine features. And if you look over here on the lower right, you can see the classification of how these tumors go from an AR positive to an AR negative state and some of the molecular markers that you might look for. And some of the common ones are P53 mutation, RB deletion, as well as gain of function of chromograninase, synaptophysin, and one of the proteins we'll discuss in more detail is RET. So back when I was a postdoc at UCLA, one question we always asked was, the mutation rates of prostate cancer is actually quite low, especially in like druggable genes. So we went off to ask a question, can we identify other markers that may predict or may help find new targets for this late stage disease? So we did a very simple experiment where we used 4G10, which is a phosphotyrosine-based antibody, to ask, do we see other modifications in these tumors beyond just the genome? And here we looked at the phosphoprotein levels. And what we found is that in castration-resistant prostate cancer, we saw a significant increase in staining intensity of phosphotyrosine signal compared to hormone-naive or high-grade prostate cancer, which is localized. So we published this, over a decade ago now, which I can't believe it's been over a decade ago. And what we thought was, if we can find some activated kinases that may be driving the biology, maybe we can find some new druggable targets in this, castration-resistant state. So we published a lot of this work, but what we didn't know is, can we also find these activated kinases in aggressive variant prostate cancer phenotypes, such as neuroendocrine prostate cancer, double negative prostate cancer. So we developed a phosphoproteome approach. I did this back in my postdoc days, but in general, it's a very simple strategy where you can take, tissues of interest. Here we do cancer tissues, you can do cell lines, you can do other tissues of any type that you really can imagine. We lyse, homogenize in a 9-molar urea, digest with trypsin, and then we purify our peptide mix. If you want to look at the phosphoproteome, which is what we'll call this, you need to do an enrichment step, because if you just took this pool and ran this on the mass spectrometer to identify these peptides or these phosphopeptides, you'd mainly identify non-phosphopeptide components. So we have to do enrichment steps. So the enrichment steps we typically do is a phosphotyrosine-based enrichment where we can take 4G10 and do a simple pull down to pull down these phosphotyrosine peptides. And then we do a simple enrichment to remove any further non-phosphopeptides by doing titanium dioxide. So titanium dioxide will just further enrich for phosphorylated species, independent of whether they're tyrosines or serines or threonines. And then we get an enriched pool here, that's our phosphotyrosine pool. We can clean that up and we can run it on the mass spec. And in parallel, we can do this with the serine threonine peptides or phosphopeptides, and by taking the supernatant after our pull down. We can fractionate if we want more coverage. And then we do titania here again to remove any non-phosphopeptides that may have carried through, clean it up, run it on the mass spec. So in parallel, you can get both phosphotyrosine peptide data as well as phosphoserine threonine peptide data, depending if you're interested in tyrosine kinases or serine threonine kinases. So as Kaylee mentioned, I've been highly collaborative in this work, and we've worked with a lot of groups to do this, both within my lab and also outside. So these are some of the papers that have kind of used this technology that my lab has helped to work with, including some of the work down here at the bottom that I'll discuss with you throughout this talk. And then here's just some of the papers that we did in prostate cancer itself that I did as a postdoc and currently now as a professor here at the University of Minnesota. We use mouse tumor models. We've used rapid autopsy in patient samples. And this paper here, authored by Megan from the lab, just came out a few days ago. And a shameless plug, if you want to go see her presentation in October, go to the Tango meeting, which is the third Thursday, right, Justin, at 3 o'clock? Yes, all right. Come on out and see that work. And so what I'm going to show you today is a lot of the work that we just published looking at prostate cancer-derived xenografts. And this is the LUCAP series from the University of Washington in Seattle. And then I'll also talk to you a little bit about cell line EV proteome, as well as plasma-based EV proteome from patient samples. So the LUCAP PDX series, this is developed by Eva Corey at the University of Washington in Seattle. And basically they published a paper about 5-6 years ago now, characterizing these LUCAP PDX tumors. And what they're able to show is that the LUCAP PDX tumors, both at the amplification and deletion level, so genomically, look very similar to prostate cancer patients, shown here. So the reds are the amplifications, the blues are the deletions. And then also through IHC staining, you can see different protein markers that are either upregulated or downregulated in these PDX tumor models. And what they find is that there's a cohort here that actually have neuroendocrine phenotypes, shown here on the right, that lack some of the AR proteins, but gain some of the neuroendocrine proteins like synaptophysin and chromogranin A. So as you can imagine, these PDX tumors have been widely used for multi-omic strategies. So from genomics to RNA-seq to metabolomics. But really what has not been done yet on this cohort of tumors is the proteome and the phosphoproteome. So we worked with Eva Corey and Steve Plymate at the University of Washington to get these samples and actually run them and develop a resource for the community to have a proteome and phosphoproteome database to add to the rest of this omics. So Zoe in the lab helped work on this project. So she was able to take 6 adenocarcinoma PDXs, 6 patient-matched adenocarcinoma castration-resistant PDXs, and 6 neuroendocrine prostate cancer PDXs. And she was able then to run in triplicate the proteome and the phosphoproteome of all of those tumors. All right, so here's just a characterization of the number of proteins and phosphoproteins that we were able to identify. And then also here is just kind of the workflow. And again, the workflow strategy is very similar to the strategy we used on the previous slides that I showed you earlier. So as you can imagine, the adenocarcinomas, these PDXs, cluster uniquely away from the neuroendocrine PDXs based on the proteome. So here is just shown a hierarchical clustering heat map. This is unsupervised clustering. And you can see here also that if we select a certain number of proteins that are enriched in the adenocarcinoma tumor, shown in blue, we see AR, FOXA1, all the standard protein markers you would expect to find in prostate cancer. And then if we look on the lower bar graph, we find that the orange bars are the ones that are enriched in the neuroendocrine tumors. So you see enrichment of chromogranin A, CCAM5, synaptophysin, et cetera. So again, we find the common features at the protein level, both for adenocarcinoma as well as for neuroendocrine prostate cancer. And this is just a heat map showing the differences of these proteins in a visual form instead of a bar graph form. So in the phosphoproteome, we kind of see the similar phenotype, although there is a little bit of blending in of these different tumor phenotypes. And what's kind of interesting is there's a subset here of adenocarcinomas at the phosphoprotein level that blend in with the neuroendocrine tumors, right? We don't really know what that means just yet, but what it could suggest is that there's some common features of signaling that may be common in these adenocarcinoma tumors that are similar to the neuroendocrine tumors. And hence, maybe these tumors are the more advanced adenocarcinomas that may be progressing towards a neuroendocrine state, all right? Don't know that, we need to test that. And again, we can take some of the phosphopeptides or phosphoproteins that are enriched in both the neuroendocrine tumor cohort or the adenocarcinoma cohort, and we find some very common proteins and phosphoproteins that we would expect to see in these different cohorts. And then finally, on a volcano plot over here, we can show that we see some enriched phosphopeptides in the neuroendocrine cohort and the adenocarcinoma cohort. And what's cool about this list here on the volcano plot is these are functional phosphopeptides. So all of these phospho-residues that are shown here on these volcano plots actually have a known function, all right? So if you want to really look for the function of some of these proteins, you might get a sense for what's going on between these two different phenotypes. And what you notice one here is red. So again, kind of lends into the story in the second half of the talk. So just briefly to summarize, this is published work, so we can dig into these, and I'm happy to share the Excel tables for these PDX models if anyone wants them to dig into that in more detail. So we've created a first and largest PDX proteome phosphoproteome data set. These protein and phosphoprotein expression correlates to known markers of adeno and NEPC. All right, so that's great, but really, what about the proteome from liquid biopsies? Because that's really where we want to move, is to be able to do this not from tissues, but actually from blood, plasma, or urine. So as you can imagine, if you want to get analytes or proteins of interest from a patient, the more aggressive the tumor, the easier it is to find these proteins or to find these analytes, all right? The earlier stage of the tumor, the harder it's going to be. So it makes sense that the sensitivity needed for detection goes down as the tumors become more aggressive from left to right. And it makes sense that the increase in circulating tumor content goes up as a tumor becomes more aggressive, and it goes down as a tumor is less aggressive, all right? That's known, that's been published both from CTCs to EVs to circulating tumor DNA, all that is pretty well established. So as you can imagine, if you get a tube of blood from a patient, you can basically look at several different analytes that of interest: circulating tumor DNA, cell-free DNA, methylated DNA. You can look at microRNAs. You can get extracellular vesicles where you can look at DNA, RNA, or protein. And even if you want to look at the cells, you can enrich for the CTCs and look for the DNA, RNA, or protein from the CTCs, okay? So we're kind of trying to hit several different angles for using these approaches, and I'll kind of give you some snippets of each of those as we go through this. So the first one we're actually looking at is from extracellular vesicles. So basically these are, basically lipid droplets that are actively secreted from cells. It's thought that these contents, can contribute to signaling and to other mechanisms between cell types in the tumor microenvironment. So cancer cells may secrete exosomes that then work on macrophages or other stromal cells within the tumor microenvironment, and it may be reciprocated and go back. All right, so they're very abundant. So that's why we actually think they might be useful for us as an approach to look for biomarkers. But the challenge that we've had up until now is that the EVs that are released are really not cancer specific, all right? Normal cells pump out EVs, cancer cells pump out EVs. So you get this mixture of normal and cancer in the blood, and trying to differentiate the two is actually very challenging. So, I was kind of skeptical about EVs probably 3-4 years ago because we couldn't get consistency in detection of these EVs. But I think what we wanted to do is start to see if we could develop methods to find these EVs and consistently find proteins across the different sample types. So Megan really took this on, and we started in cell lines first, because if we can't find consistent proteins in cell lines, we're not going to find it in patient samples, all right? So Megan, what she did is she took a panel of AR positive cell lines, AR negative cell lines that are both double negative, as I mentioned before, and the AR negative neuroendocrine positive, which are the neuroendocrine prostate cancer cell lines. All right, she enriched for the EVs through several rounds of ultracentrifugation. All right, so you can imagine this requires a lot of spins and a lot of time. You get these enriched EVs. You then take those EVs and isolate the protein and develop peptides similar to the methods I described to you before. You run it on the mass spec, and then you get your list of proteins that you have identified. So Megan ran these in triplicate, and I'll show you the data here in a second. So first, we were able to successfully enrich for EVs, all right? So we get EVs at the proper size, between 1 to 200 nanometers, all right? So that's pretty typical. And then the EV markers that we find are actually enriched in our EV preps and not in our whole cell lysates. So 2 EV markers, ALX and CD9, are enriched in all of our 3 different cohorts, and we don't really see them in the whole cell lysates. And then a cell lysate marker is found in the cell lysates, but not in the enriched EVs, all right? And again, suggesting we're actually enriching for these EVs. So when Megan ran the proteome, she was able to get at least 2,000 proteins across all of her different sample types and 4,000 proteins across the whole cohort, so that's great. And we're improving that actually on a daily basis. And then what was really striking to me, and I was kind of surprised by this data, and maybe looking back, it's not that surprising, is that you could actually cluster them by subtype, all right, just by doing the EV proteome. And I thought that was pretty astounding. So we're able to show that the AR positive cell lines all cluster together, the double negatives cluster together, and the neuroendocrine tumors cell lines all clustered together, all right? And we can show this here in an unsupervised hierarchical clustering heat map, where again, they cluster uniquely away from each other based on the different subtypes, all right? So that was pretty cool, all right? So that shows that we can get some consistency across these preps, and we can find these proteins that may segregate these different subtypes out. So Megan went a little further and looked at AR activity and AR cofactors, and found that in blue here, which are the AR positive cell lines, they had higher AR activity scores in the double negative or neuroendocrine tumors. This is just a heat map of some of the proteins that we found where these AR cofactors are, as well as another heat map of some of the AR cofactors. These are the AR activity genes, and these are the AR cofactors. We were also able to do that on the neuroendocrine prostate cancer cell lines, and show that we had a higher neuroendocrine score with the neuroendocrine cell lines compared to the others. And again, this is just a heat map showing those proteins as well, showing enrichment of these neuroendocrine genes. And then finally, we're able to show that the double negatives carry basal cell and EMT proteins, all right? which is somewhat expected from the literature about what double negative tumors are and how they behave. So here in red are the two double negative cell lines, and they have a higher basal signature compared to all the other cell lines. So that was all fantastic. So we could actually cluster and subtype each of these out based on just the EV proteome. And then finally, what I thought was, quite interesting is that some of these different proteins are actually differentially expressed across the different subtypes. So a few that you would expect, so you see PSMA enriched in the AR positive prostate cancer cell lines along with the STEAP proteins, STEAP1 and STEAP2. You see enrichment of NCAM, DLL3, and CCAM in the neuroendocrine prostate cancer cell lines shown in green. And then what was kind of interesting is you see TROP2 enriched in the double negative prostate cancer cell lines shown in red, all right? So again, we can kind of find, unique druggable proteins that may differentiate these subtypes as well. All right, so that's great. Cell lines, very controlled. All right, what about the EV proteome in patients? And that's where it gets really challenging, and I'm going to show you some of that data. So we worked with Dr. Emmanuel Antonarakis and Dr. Ali Arafa to partner on a clinical trial where we use Pluvicto, which is a PSMA radioligand targeted agent, in castration-resistant prostate cancer patients. And here we have a trial of 100 patients. So why are we moving to this trial from the cell line data? Well, one is PSMA-based therapies. There's very little predictive biomarkers for response to that therapy, right? Typically what's used is a PSMA PET scan to enroll the patient onto the trial, but then that patient may or may not respond based on the therapy itself. So we need to figure out if we can find a better biomarker for those patients. And then two, can we consistently find proteins across these patients, of 100 patients, that may help identify responders from non-responders, okay? So those are kind of the two goals of the trial. So why PSMA? Well, it's highly abundant. It's prostate-specific membrane antigen. It's linked to higher Gleason score, poorer survival outcomes. And the US FDA has approved the use of PSMA PET scans for better treatment planning and targeted care in metastatic prostate cancer, all right? And that's what's used to actually enroll patients onto the Pluvicto trial that I'll show you here in a minute. So how did we get to this point? Well, the VISION trial headed by Dr. Oliver Sartar at the Mayo Clinic had a trial where he used Pluvicto to show that you could get improved survival and progression-free survival in patients on PSMA radioligand therapy, all right? But the problem, again, is that 30 to 40% of patients don't respond to this treatment, even though they're PSMA PET positive on their PET scans, okay? So we still need to find a better biomarker that may predict these patients responding and not responding to therapy. So there's been some studies out there that have looked for particular biomarkers. These have typically been at the genomic level, all right? So there's a couple papers here done by Dr. Sartar and others where they found FGFR1 and a cyclin protein, cyclin E, as well as P10RB and P53 alterations from circulating tumor DNA that may be predictive of response to Pluvicto in patients. All right, but no one's really looked at the protein level, and I'll get to that in a second. And then Dr. Andy Armstrong at Duke also was able to show that PSMA positive CTCs increase on patients who develop resistance to abiraterone and enzalutamide, again suggesting that we see an enrichment of circulating tumor content in patients that are developing resistance to therapies that may be potential candidates for Pluvicto itself. So here's the study design of this particular trial. So we did a 3 time point trial. So we get 3 time points per patient. We have a baseline collection, and then we have a collection at dose prior to dose 3 of Pluvicto, and then a final dose at progression. All right. So what we do at each of these particular time points is we do CTC enumeration for PSMA positive CTCs. All right. We do exosome proteomics, and this is done by Ali in the lab, where we look for EV proteins that may be predictive of response. And then with Dr. Scott Dem, we do circulating tumor DNA to analyze both genomic features as well that may be predictive of response. And we do that for all 3 of these time points if we're able to get them. So I'm going to show you a snippet of just the first quarter or so of this trial. And these are the patient characteristics of the first 27 patients. So you can see here that most of these patients are in grade groups 4 and 5. And the majority of these patients have bone and lymph node metastases, all right? So these are aggressive patients with multiple metastases in these patients, all right? So what Ali did is he had to first develop an extracellular vesicle isolation protocol that would be consistent to find these proteins. So this is what he did. So he basically went through several rounds of ultracentrifugation, so very similar to Megan's protocol. But the main difference that he did is he had to do what was it, 4 or 5 ultracentrifugation spins and washes to clean this up. Okay, and why did he have to do that? Because there is tons of serum in albumin and other contaminating proteins that would basically take up all of the mass spec runtime, and we would find nothing else. So we do several rounds of spins and washes to clean that up. All right, and that's really what fixed this whole problem. Until then, we were never able to really get anything of interest through the EV proteome. So that's really what happened. And I'll show you the data here on what we found. So before we get into the proteome data, what we do find is that routine clinical data does not predict treatment response in these 27 patients. So we were able to get about 12 responders and 15 non-responders, and the responders have a greater than 50% PSA reduction, all right, compared to baseline. That's what's considered a responder in this particular case, all right? So baseline PSA does not predict it, baseline PSMA PET scan mean, and baseline PSMA PET scan max does not predict responders from non-responders in these cohorts, all right? We don't see any difference in EV markers, all right, but we find them, so that's great. All right, so we find CD9, CD81, and CD63 markers, all right, but they don't predict response and non-response in these patients, but we consistently find them. So I think that's a testament to the consistency of these proteins that we're able to find. But what we do see is that PSMA protein from EVs actually is predictive, all right? And some of you might be like, well, okay, that's obvious, but it's not that obvious because the PSMA PET scans don't, okay? So it's not like that black and white. But what we're able to show is that the responders have a much higher level of PSMA protein in their EVs at baseline compared to the non-responders. And we can break this down into exceptional responders, which is PSA reduction greater than 90%, or the standard PSMA 50, and non-responders. And we see kind of a gradient of response based on the levels of baseline PSMA protein. And then finally, we can also see a very nice correlation of PSMA protein to PSA progression-free survival, showing a very nice correlation across these patient cohorts that are responders. So what we think we're finding is that maybe PSMA protein from EVs actually is acting as a very solid predictive biomarker to patients who may respond to Pluvicto. And then finally, we looked at just a few other interesting proteins or targets of interest. And what we find is that they actually are not predictive of response, all right? So PD-L1, STEAP4, and B7H3, all of these are druggable proteins in the clinic for other cancer types, and none of these proteins from the EVs actually predict responders from non-responders in this particular trial. All right, so to summarize the EV proteome, so they contain important information about phenotypes of cell of origin, and the protein markers in EVs have the potential to differentiate specific prostate cancer subtypes. So patients with higher EV-derived PSMA expression had better treatment response rates compared to patients with lower PSMA. So I showed you that data a couple slides ago. And the PSMA-based proteome from EVs predicted treatment durability on Pluvicto with patients who are on Pluvicto. So we think that these exosomal-based proteomics may be able to select patients who would benefit most from treatments in trials. And this PSMA trial is just one example of that. And we're going to continue finishing up this trial with the remaining patients. We've recruited about 90, give or take, right now. So we'll be doing the full cohort later this year, and hopefully by next year we'll be able to update you on where we are with that completed trial. All right, so on the second half of the talk, I'm going to switch gears and tell you a little bit about RET kinase, because that's one of the main proteins that we study in the lab. And this is one of the papers that was published a few years ago that really implicated RET kinase in neuroendocrine prostate cancer, and I'm going to show you some unpublished data on how we've carried forward this paper and into the mechanisms of RET signaling and what drives RET. So this all started when I was a postdoc at UCLA, and this was an N of 1 patient as well. So this is a patient where we got rapid autopsy material from Ken Pienta when he was at the University of Michigan at the time. And we were able to get multiple metastatic lesions from the same patient. In this case, this patient had small cell neuroendocrine carcinoma, all right, which is the same thing essentially as neuroendocrine prostate cancer. And what we found when we did a phosphotyrosine array is that RET tyrosine phosphorylation was elevated in this patient, all right? And when I first saw this, my initial thought was, well, that's kind of interesting. RET's a neuronal driven gene, All right, it makes some sense that it's expressed there, but no one really knew anything about why it's expressed and what's going on in that patient. So that led to the first paper that was published a few years ago. And this is data from that paper where we're able to show that patients with neuroendocrine prostate cancer phenotypes, shown here in red at the bottom, have very high levels of RET and ASCL1 and low levels of the androgen genes such as PSA, AR, and NKX3.1. All right. And then in green here are your AR positives, and we see very little RET gene expression. Now, I've been asked this question before, and I still don't have a good answer for you. Why do we see RET expression on some of these AR positive tumors? And we don't really know that and why that is the case. We postulate these are tumors that are in transition, or maybe it's a tumor that has an adeno and a neuroendocrine on the same tumor. All right, that's very possible. So we don't know, but we'll keep investigating that. So what is RET? It's a receptor tyrosine kinase. It's driven mainly through its ligands. GD and F are the ligands that drive it. And the downstream signaling components are very standard and very similar to a lot of RTKs that you would be interested in, such as MAPK signaling and AKT signaling. All right, so that makes a lot of sense. And that's what you typically see in RET as well. All right. The unique thing about RET is it's primarily expressed in neuronal cells and required for neuronal development. All right, and that's kind of why we think it might be an Achilles heel in these neuroendocrine tumors, because the machinery that's driving these tumors may be trying to push those tumors into kind of a neuronal state. And I'll show you some of that data here as we go forward. There's activating mutations in this gene, all right, and they typically drive the formation of tumors in neuroendocrine-like tumors, such as MENS or multiple endocrine neoplasia type 2, papillary thyroid carcinomas, and certain lung cancers, all right? And there are 2 RET-targeted drugs that are out there now that are approved essentially for any tumor that has RET fusions, all right? So this is blue and Loxo, which is pralsetinib and selpercatinib, all right? Problem is we don't see RET fusions in prostate cancer, so we have to kind of think outside the box a little bit as to what patients may be targeted in this cohort. So this is a huge collaborative effort from different people in the lab, some of which have moved on to other positions, and some of that are currently in the lab, such as Sachi Chow and Sasha. And what Helena initially did was take AR positive and AR negative cell lines and run the phosphotyrosine proteome on these particular cells. And we're able to show, this is the RET receptor tyrosine kinase signaling pathway, is that a lot of the AKT and MAPK signaling components are actually hyperphosphorylated in these neuroendocrine tumors or cell lines compared to the AR positive cell lines, all right? And a lot of these phosphosites shown here in yellow, so yellow is hyperphosphorylated, are actually activation sites of these particular proteins. So this hints to us that the RET pathway is indeed activated in these AR indifferent or AR negative neuroendocrine cell lines compared to the AR positive cell lines. So we took a neuroendocrine cell line known as NCI H660. This is a neuroendocrine prostate cancer cell line. They express high levels of RET protein. We can knock down RET protein and show we can basically perturb the growth of these cells over time. All right, and we have some in vivo data I'm not going to show you because all this is published, and I want to get into the new stuff. So that's great. So we've been able to implicate RET in neuroendocrine prostate cancer and think it might be an Achilles heel for treating patients who are RET positive. But we still don't know what drove RET to this particular phenotype, all right? So that's where a lot of this work has come in, led by Sachi Chow and Sasha. So what we're able to show is that RET can actually correlate with ASCL1, and I have a lot of data kind of implicating that. But basically where it all started, it was this paper from 2021 that took PDX models and was able to find that the PDX NEPC tumors can be classified into 2 main subtypes based on the transcription factors that are expressed. One is a NeuroD1 subtype, and the other is an ASCL1 subtype, all right? And what we're able to show is that RET is more correlated to ASCL1, and I'll show that on the next slide. But the initial data that got us to thinking the ASCL1 RET connection was this data published by Amina Zubidi a few years ago, where they took the H660s and did an siRNA knockdown of ASCL1 and showed that RET was one of the top cell surface genes to actually be reduced when you knock down ASCL1. All right, so there's a very high positive correlation to knockdown of ASCL1 and loss of RET, all right? So we went into the cohort of tumors. Here we have The LuCap PDXs, these are the same PDXs I described to you earlier in the talk. And you can see here, oh, I'm sorry, these are not the LuCap PDXs. These are the Lee Ellis single double knockout and triple knockout. These are P10 and RB double knockouts, or P10, RB, P53 triple knockouts. The double knockouts and the triple knockout mouse models generate neuroendocrine prostate cancer. All right, so that's shown here in green. And what we're able to show is we see a very nice correlation between RET and ASCL1 in the green tumors, which are the neuroendocrine tumors, compared to the AR positive adenocarcinoma tumors, which are simply the P10 knockout tumors, which are AR positive. On the right side, when we compare neuro-D1 to RET, we see no correlation whatsoever. So again, this kind of suggests that we see kind of RET, ASCL1 connection, but not neuro-D1 and RET. So if we look at actually patient samples, so this is the rapid autopsy series from the University of Washington, and this is another cohort from Misha Beltran's group. In green here are the neuroendocrine prostate cancer samples, and we see a very nice correlation between RET and ASCL1. On the right with neuro-D1, we don't see that correlation to RET. And then on down here on the bottom, the red or the NEPC tumors, we see a very nice correlation to RET and ASCL1, and we don't see a correlation to RET and neuro-D1. So again, correlative evidence suggests this RET-ASCL1 connection exists. We even moved to the single cell level, where we were able to work with Dr. Justin Wong's lab to take single cells from about 12 to 14 different patient tumors and ask, do we see at the single cell level expression of RET and ASCL1 in the same cell? And what we're able to show is indeed that's the case. So here is in blue are the NEPC tumors shown in the bottom part. They have an NEPC signature, all right, whereas the adenocarcinoma tumors or the AR positive tumors do not. Most of those NEPC tumors express RET, and the ones that express RET also express ASCL1. The one that's RET negative expresses neuro-D1, all right? And we can show here on a kind of a dot plot where the bigger the dot, the higher the number of cells express that particular gene. We see a very nice correlation to RET and ASCL1, but we don't see that correlation to neuro-D1, all right, at the single cell level. And then finally, we're able to look at this temporally. So we took a model that was developed in Owen Whitty's group. These are very standard human recombination models. In this particular case, they took 5 genes where they did a double negative P53, or dominant negative, I'm sorry, P53, activated AKT, knocked down RB1, overexpressed c-MYC, and overexpressed BCL2. You make virus for all of those, put them all together in one cocktail, and you can infect prostate basal cells from a patient. And over time, those tumors, when implanted into a mouse, will form tumors. And what you can get is, with this combination, is a neuroendocrine prostate cancer phenotype. All right, and that happens out of the course of about 12 to 14 weeks. So this just kind of shows what the PARC-B xenograft looks like and a human small cell prostate cancer, so they look very similar histologically down here at the bottom. So as you can see here, what they did is at different time points, they took these tumors out and did RNA-seq. So that was very beneficial to us because we could just take the data and ask, can we temporarily see expression of RET and ASCL1 in these tumors? And quite strikingly, starting at about time point 3, which is roughly when these tumors start to develop neuroendocrine features, RET and ASCL1 start to increase in gene expression. And they do it concomitantly, which is quite striking and pretty amazing, actually. And as you get to time points 4, 5, and /6, which are kind of the late stage neuroendocrine tumors, you kind of see the max expression of a lot of these two genes. So what we're seeing is that when ASCL1 gets turned on, RET also gets turned on, all right? Now the question is it RET ASCL1 or is it ASCL1 RET? And we'll get into that in a second. And then finally, we were able to show with Dr. Justin Wong's group using their technique called ALAN, which allows you to look at gene association networks and start to ask, what other genes are in close association with RET or ASCL1 in these tumors, both in adenocarcinoma or in NEPC. And what we're able to show is that in the adenocarcinoma tumors, all right, when we look at RET, we see very little association of a lot of these neuroendocrine transcription factors. Okay, it's kind of expected. But when you look at the neuroendocrine tumors, we see a very high association of RET and a lot of these neuroendocrine transcription factors, suggesting that RET and all of these neuroendocrine factors are all in close networks, close association with each other in the particular tumor. We can show this visually here in D&E, where we'll look at the RET signature, and we find that in the adenocarcinoma samples, we don't really see a high correlation at all with any of these genes, but we see a very high correlation in the NEPC tumors. And we can reverse it where we use ASCL1 as the common feature and look for transcription factors that may be associated with ASCL1. And we see very little features associated with ASCL1 other than FOXA1 in the adenos. And we see a lot of these transcription factors associated with ASCL1 and NEPC, with the exception of NeuroD1. All right, so again, this kind of dichotomies is still holding true in these samples as well. All right, so lots of correlative evidence. All right, is there any direct evidence for this? So we dug into some ChIP-seq data and kind of asked that question. And we're able to show is that indeed, yes, RET does bind to open chromatin on the RET promoter and enhancer region and drives expression of RET gene itself, all right? So this is H3K acetylation ChIP-seq. And here we have ASCL1 positive cell lines and PDXs and NeuroD1 positive PDX in blue. And what you can find is that at the RET locus, we see high open chromatin, so a lot of acetylation on the ASCL1 positive tumors and cell lines. We do not see that in the NeuroD1, all right? When you do ASCL1 ChIP-seq, we find that ASCL1 binds that open chromatin on the ASCL1 positive cell lines and LeuCap tumors, but we don't see that in the NeuroD1 positive PDX, all right? So again, this suggests that ASCL1 binds to this RET enhancer promoter region and drives gene expression of these genes. I don't have the RNA-seq data on this, but RET gene expression is actually quite high in all of those ASCL1 positive tumors. And then we can also show this using the LNCap model. So again, kind of the same concept where we overexpress ASCL1 and LNCaps compared to a control. And again, we find that ASCL1 binds to the open chromatin on the RET locus, and we get enhanced expression of RET here in purple, all right, gene expression. And like I mentioned before, due to time, I don't want to show all the lung data, but we have a lot of lung cancer data kind of showing this very similar phenomenon where RET and ASCL1, where RET is driven by ASCL1 in a panel of both lung cancer cell lines and lung cancer tumors. So we're seeing kind of a pan-cancer effect of this RET ASCL1 axis in these particular patients. And then finally, all right, which comes first, the chicken or the egg, RET or ASCL1? So we knocked down ASCL1 and RET in H660 cell lines and was able to show that if you knock down ASCL1, all right, RET protein goes down. If you knock down RET, ASCL1 protein does not go down. So it makes sense. ASCL1 is upstream of RET. It controls RET function and RET fate, all right? And here we just quantifying some of the differences there. All right, so some conclusions on the RET ASCL1 axis. So RET is overexpressed and targeting RET reduces NEPC tumor burden. We published this paper a few years ago. It strongly correlates to ASCL1, but not the NeuroD1 subtype in neuroendocrine prostate cancer as well as in lung cancer. It shares highly similar gene networks in NEPC, but not in adenocarcinoma. And ASCL1 binds to RET regulatory sequences and regulates its expression, both at the RNA and seems to be at the protein level. So where do we want to go next? We have a lot of questions that we still want to answer here and a lot of projects that are ongoing. So we know ASCL1 drives expression, so how do we target this loop? All right, we know ASCL1 is phosphorylated. Okay, we have data and evidence from the LeuCap PDX tumors that ASCL1 is phosphorylated on 2 unique sites. Nothing is really known about those phosphosites. So we might dig into those and actually ask, what's the function of those ASCL1 phosphorylation residues? Look for other kinases and proteins that are regulated by ASCL1 from these existing data sets, and one we found was KIT. And if for those of you in the field know that KIT is actually involved in late-stage prostate cancer, it's one of the key targets. So that's kind of interesting to see that there may be another target of ASCL1. We also know that non-kinases like DLL3 is also regulated by ASCL1, and that's already a druggable target in the disease. All right, can these tumors on RET inhibitors be sensitized to other therapies that are currently used in the clinic? All right, that's an open question, something that we can go and test. Can we develop, biologics to RET? All right, so both of these are active in the lab. So we're working with Dr. Dan Harkai to look at PROTACs, and we have some funding actually to do that. All right, so protein degraders. And then we're just starting up a project to develop RET antibody drug conjugates where we have some unique scFvs that bind to RET, and we're going to develop these into ADCs with Dr. Harkai. And then finally, can RET protein be used as a biomarker for treating neuroendocrine prostate cancer by using CTCs or liquid-based approaches like extracellular vesicles? And I'm going to go on the last couple slides here, just kind of showing you our approach to this. So this is a DoD-funded project where we're actually trying to identify RET in liquid biopsies. So here is a cohort of 50 patients. These are metastatic castration-resistant prostate cancer patients. This is in collaboration with Dr. Antonarakis as well. And what we do is we take 50 mils of blood, and here we're going to enumerate RET-positive CTCs, and then also take the CTCs that we isolate and perform targeted proteomics on this to look specifically for RET protein. And we're going to do this at baseline, and then the patient's going to receive a physician's choice of therapy, and then we're going to collect another blood sample at progression. All right, so that's kind of the idea, and the hope is that we see increased RET gene expression at progression, because that's typically what we see clinically. All right, so we've developed some RET peptides. So this has been done by Chow in the lab. We have about 5 or 6 RET peptides we've identified. We're able to pick them up on the mass spectrometer. And here is just an example of one of these heavy and light labeled RET peptides, where we can see very nice peaks and very nice transitions of both the heavy labeled and the endogenous RET peptide in these samples. So working with Astron Biosciences, we're able to then enrich for these CTCs using bead-based technology and holographic imaging. And when we can isolate those CTCs, then we can actually perform targeted proteomics. And this is still a work in progress. This is by no means ready for prime time, but we're getting very close to this. And this is just a snippet of some of the proteins that we can pick up from CTCs in patients. So after we enrich for the CTCs, we can look at these panel of prostate-specific proteins and look for expression or non-expression of these, and we're going to likely add RET and other proteins to this list as well. All right, so to summarize everything up, okay, a little biased, but we feel that kinase activity is a critical Achilles heel in the treatment-resistant prostate cancer. Okay, these are typically in states where AR is lost or AR is indifferent. All right, so not in your AR-positive tumors. So identifying those pathways will be critical because we can't really find it genomically. These mutations don't really exist. We think using a proteomic strategy or a phosphoproteomic strategy may be the key technology to uncover this. And once they're found, I highly doubt that targeting just that kinase is going to be efficacious. We're probably going to need to think of co-targeting strategies. What that is, I think, is still to be determined. And then, can we use liquid biopsies can be the source of material for this analysis. So instead of taking tissues, we can just take blood or urine and look for these particular markers. And then they may help to predict treatment response. So I'll end there, and this is the most important slide because these are the people who did all the work. I'm just their cheerleader. So Megan and Ali did a lot of the cell-free work or the liquid biopsy work. Shashi, Chow, and Sasha did a lot of the RET ASCL1 work. A lot of people have contributed to those projects that are currently off to bigger and better things, I like to say. And I'd also like to thank the mass spec core facilities. You guys have been fantastic in letting us use those machines and run our samples. Without you, we wouldn't be able to get any of this awesome data. And then the funding sources, the NIH and the DoD, they've been very, very generous to let us, kind of go outside the box a little bit to run some of these projects. And then finally, a plethora of collaborators on the RET kinase project, including a lot here at the University of Minnesota, Scott, Justin, Emmanuel, and Dan. Without your help, none of this work would have been able to be accomplished. So with that, I'll take any questions. Thank you for your time.
Raise your hand. I've got microphones if you've got a question.
So I got a question of the EV proteomic study. So is most of the protein detecting in this study is the cytosol protein? Is it both cytosol protein and the DNA binding protein?
So is it mostly cytosolic proteins or DNA binding proteins? Maybe Ali can better answer the compendia of proteins we're finding. I would say we're not finding a lot of DNA binding proteins. So typically the mechanism of exosome release usually contains a lot of cytosolic or cell surface proteins. So there's going to be the majority of proteins that we find. We may find here and there a few DNA binding proteins, but I'm skeptical. Like an ASCL1 assay would be something I would think about from exosomes because I doubt we would find it. Do we pick up AR once in a while? Yeah, so even AR is a little iffy, and these are, that's a highly expressed protein. So I think it's probably borderline that we'd pick up a lot of these DNA binding proteins. I think you'd rather focus more on cell surface or cytosolic.
Thanks.
Yeah.
Hang on.
Yeah, thank you. That was really interesting. I'm wondering, especially going back to that heat map you showed where ret was on where it shouldn't have been, and you're like, maybe these tumors are like trying to become neuroendocrine. And I thought, and I don't know anything about prostate cancer, so maybe this is naive, but I mean, could you maybe use your approach as like to catch these guys early on and then think about co-targeting therapeutics that like prevent them from turning neuroendocrine and nasty? Is that a reasonable sort of strategy at some point?
It's reasonable. I think it depends on finding the right marker and also, what's the frequency of those tumors, in the clinic, right? If it's 1%, it's going to be really hard to do a study like that.
Form, right?
Yeah, those are rapid autopsy specimens, so keep that in mind. So it's hard to know when that transformation happened. The RET study that we currently have funding for may help provide some insight into that because the patients are going to we're going to get a baseline sample, and then they're going to get physician's choice and then another sample. And that physician's choice typically is going to be your second generation hormonal therapies. All right, so we might get some clues as to what the actual baseline levels are, at least of RET protein, because that's what we're going to look for, and see what that frequency is, and then maybe we can start moving further up into that spectrum. But we'll see what that data looks like first before I can postulate anymore. So.
Yeah, really nice talk. I had a question with regard to RET, a couple of questions. One is, what's the normal tissue expression levels relative to what you're seeing in the cancer cells, and where is it expressed? Is it neurally expressed? Is that where it's expressed, or?
Say, is it neurally expressed?
Yeah, is it expressed in is it expressed in CNS basically, or?
Yeah, it is, and and that's why a lot of the target therapies to RET are in neuronal driven cancers because that's where they're typically derived from. So yes, that's where the normal expression is found. I don't know the relative levels of RET gene expression in cancer versus normal. We should probably look at that. But
okay,
if ASCL1 is turned on, it's going to drive robust levels of RET. We see that in small cell lung cancer. We see that in prostate cancer. And we don't see that in the adenocarcinoma tumors, all right, at least at the protein level and the gene expression level. But how it compares to the normal prostate, it's higher. I just don't know what the absolute numbers are.
And the GFR alpha that was up there, what's the association with RET? Is it always associated with it? If you find RET, do you find it nearby, or?
The co-receptor?
Yeah.
Yeah, you need the co-receptor to activate it. So you need the ligand and the co-receptor. So you need both of those components to help RET dimerize and become activated.
All right, thanks.
Mm-hmm.
That was a great talk. Thank you for sharing your work. I think I had a similar question to Rick, actually. The PROTACs and the ADCs are really exciting, but do you think there are going to be effects in the neurons if they really need RET for function?
Yeah, great question. That's, I put in a RET degrader or a RET ADC project to the DoD, and that was the main critique. Okay. So don't have a great answer for you other than the RET kinase inhibitors don't really show that. All right, so the side effects from those drugs are typically not neuronal in nature. They're usually what you see with a standard TKI. All right, so that gives me a little hope that maybe that's not the case, but it's something we need to think about, and as we develop these, that's a possibility that could happen. Absolutely. So,
Justin, that was great. So, in the phosphoprotein part, do you look at total protein versus phospho to try to get at the biological relevance of one or the other?
Yeah, get that question a lot. There's two philosophies. There's one is if you see phosphoproteins and they're up and they're activated, you should target it independent of what the total protein is doing. All right, it doesn't matter. And then there's others who are like, well, you need to know what the total level is and the ratio to the phospho to get a sense for, is it because the tumor is expressing more protein and hence more phosphoprotein? But at the end of the day, it's more phosphoprotein, right? So I'm of the camp that if you see more phosphoprotein, it's druggable and targetable, and you should go after it.
Yeah, no.
Long story short, no.
Yeah.
But we have the protein data and we have the phosphoprotein data, especially in the LUCAP models, where we can kind of compare and contrast that. but we don't really emphasize it too much.
Yeah, I just comment in the iSpy data set, the phosphorylation of HER2 is much more predictive than the total levels of HER2. So I
Yeah.
do think that that's, if you're in terms of a HER2 targeted signal. So in second corollary from the iSpy data set, many of the prognostic and predictive things we see come from stroma and are probably not epithelial derived. And the way we're trying to get at that is to microdissect out stroma, and there's a very independent stromal signature that appears to predict response to regular old chemotherapy, but particularly immunotherapy. So have you guys done that? Can you run phosphoprotein analysis on stroma versus epithelial component?
Yeah, so can we run phosphoprotein analysis on stroma versus epithelial? The answer is yes. So for phosphoproteins, you're going to need a lot of material. All right, so that's the one downside. So, especially if you're laser capture microdissecting out these tissues, you're going to need to do quite a few serial sections, I think, to get enough material to do that. So technically you can. I think it's going to be a lot of grunt work to do it, but you'd be able to do that. And in fact, I think your assessment though is spot on. There are some papers out there to suggest that the stroma, the phosphoproteins from the stroma, contribute at least over half, if not close to 75% of the signal you may see. All right. Does that mean that the phosphoproteins you're finding are irrelevant? I don't think that's the case. It just means you may be targeting a different subtype than the cell type you think you were going after. So, but I think if you have an interest in looking at that, we should talk more because we can dig into that.
Congrats, that's a really good talk, and I especially enjoyed the fact that you only went 42 minutes and there's a lot of time for discussion, so because I think this is great. Logan criticized me at the last seminar because he said that I need to ask a question if I ask a question, but this might actually be a comment. So we've been doing EVs, exosomes, for a while, and we're looking at osteosarcoma and asking some questions that are not necessarily very different from what you're looking at. And except we're doing mRNA instead of proteins. And in our earliest experiments, we actually compared what we saw from cell lines to what we saw from xenografts. So looking at mouse serum. And the correlation was 0. There was only like a shared 2% content of RNA in those exosomes from the cell lines in a dish to the cell lines in vivo. So I don't know, maybe a question would be whether you know if the proteome might be more shared than the transcriptome in those EVs. But we developed an interesting model using xenografts that you might be interested in. It's essentially a subtractive method that allows you to look at what's coming from the host and what's coming from the donor, essentially the xenograft. And with mRNA, we have a pipeline that allows us to do that by stitching the two genomes together and mapping. I suspect you could do the same thing with proteins. And what we found is that you really need machine learning to be able to actually find these signals, because looking at the differential gene expression, you get a lot of stuff, but there's a lot of individual variation. But if you actually create some patterns and train, then the machine learning works. Mm-hmm. So I guess the question is, do you know if the proteome is more shared between cell lines and in vivo what's going on in the tumor than maybe the transcriptome is? And if you want to chat about the stuff we're doing, that guy over there, wait, Jaron, talk to Jaron.
Yeah, I mean, happy to chat, to talk about, anything proteome related, I'm happy to chat. So to get to your first question about the RNA and cell lines, so I'm guessing these are cell line derived xenografts from the same cell lines, right? Okay. So at the proteome level, that's been done, small study, and they show there are differences. So not surprising, right? You grow cells in a dish with different media conditions. You grow cells in an animal, it's going to be different conditions. They're going to get changes in the cellular signaling and pathways and networks. So that's not surprising at all that you see that. Now, the fact that you see like no correlation to all of your RNA, that's a little strange. But we do see, from that paper, I think it was at least 50%. So
the EVs were a lot more different.
Yeah. Yeah, so, and again, that's why I kind of emphasize when we did the cell line work with the EVs, that's great. And we see this very nice, connection between the subtypes, but can we see that in patients? Because that's really where the rubber meets the road. And we're starting to see that. And part of it, I think, is we just needed to clean the EVs up a lot more than what's been done in the past.
Can you get serum from the xenograft mice?
Can you get serum from the xenograft mice? I mean,
That's how we did the differentiation, is we pulled serum from the xenograft mice and then bioinformatically determined what was coming from the xenografted tumor versus what the mouse was doing. And even though you don't have an immune response, and that is very different from what you'll see in patients, it can be quite informative. There's some really interesting stuff that the host is doing in response to the tumor, and some stuff that the tumor is doing. And when you actually look at the different tumors and start looking at multiple animals, then you start seeing some really cool patterns.
Yeah. Yeah. Good point.
All right, great discussions again. And let's say thank you one more time for a great presentation.
Thank you all.